We know we use our eyes to see, but how exactly do we see? In this video, we are going to learn just that. Hi, I'm Dr. Vanessa, and this is my channel, Learning Biology with Dr. Vanessa. In today's video, we're going to take a journey through the eye and see exactly how light passes through it so that we can see and view the world around us. First thing that has to happen in order for us to see is that light has to pass through the clear portion here of the eye that is called the cornea. Now, before I go any further, if you haven't already, make sure that you take a look at my video on the anatomy of the eye because that will help you really understand the different parts that we're talking about here. So first, light has to pass through this cornea, which is this clear portion right here in the front of the eye. Now, the cornea itself is actually shaped like a dome, so it assists in helping to bend that light so that it can enter into the eye. So light will actually pass through that cornea, and then it's also going to pass through the lens, which is right back here. And if you remember from my other video, the lens has muscles attached to it, those ciliary muscles, and that helps to adjust the shape of the lens, okay? Now, what you'll notice is that depending on how much light is around, sometimes when there's a lot of light around, your pupil will get smaller, it will constrict. If there's not so much light around, it's darker, the pupil is going to dilate or get bigger. So the pupil can actually change its shape to allow different amounts of light into the eye. So the cornea is going to help direct that light through the lens, and the lens is going to change its shape to help also direct the light into the eye. And that really, again, depends on just how much light is around at that point. The lens can change the shape, and those two things are going to help get the light into the eye. Now, where do we want it to get? We want it to get to the back of the eye because that's where the retina is. That's where the special cells are that are able to convert that into the images that we see. So it first has to pass through the cornea, through the lens, which can adjust its shape, and then it's going to go towards the back of the eye. Now, as the light is entering into the eye through, again, the cornea and the lens, and it's bending, that light is undergoing refraction, which is helping to focus it on that special portion in the back of the eye known as the fovea centralis. So that area right in the back of the eye, known as the fovea centralis, is where we want the light to hit because that is going to be the area of greatest visual acuity. Now, that is part of the retina, and so it's going to hit the retinal cells in this area, and that is where we are trying to focus the light, again, for greatest visual acuity. Now, the picture that is getting sent back there is actually inverted or upside down. And then the cells of the retina in the back of the eye are going to be able to take those signals to the optic nerve. The optic nerve takes that signal to the brain, and that's how we will eventually be able to tell what we're seeing when the brain gets those signals. So now let's take a closer look at the retina and see exactly what cells are in the retina and what they're doing in order to be able to process what we're seeing so that we can actually see. Now, if we take a closer look at the retina, which is the picture that you're seeing on the screen right now, you'll notice that there are two different types of cells. We refer to them as photoreceptors, and those are the rods and the cones. Now, in order for us to see, the first step in being able to get that visual transduction is the absorption of light by photopigments that these different photoreceptors, rods, and cones have. When light hits these specific cells, a colored protein is going to undergo structural changes when it absorbs that light, and that is going to turn into a signal that is eventually going to be sent to the brain. Now, we're not going to get into super specifics in this video, but we'll talk about a couple of different things. The photopigment that we see in rods is called rhodopsin, And in cones, there are actually three different photopigments that are found there. So rods are going to respond to light, like how much light is coming into the eye, light versus dark. And then the cones is what is going to allow us to see the different colors. So rods is going to respond to the light, and then cones are going to respond and give us color vision, and that's going to depend on the different colors of light that are going to activate the different cones, again depending on color. So there are different cones, different photopigments in cones. When they get activated, then that's going to allow us to see the different colors, where rods are going to respond to light. Once the rods and cones get activated, they are then going to activate the bipolar cells, which would then activate the ganglion cells. And then these ganglion cells are going to form action potentials. The axons of the ganglion cells are going to then provide the output that is coming from the retina to the brain, and it's going to exit the eyeball as the optic nerve. So all of these signals are going to be sent to the optic nerve. Now, the way this is actually visualized is that light enters the very, very back of the retina and comes back to the front, so that when the axons of those ganglion cells go towards the optic nerve, they actually form that optic disc in the back of the eye, which is going to create a blind spot as things move out the eye. I talked about this in my anatomy of the eye video. So those axons are going to come back through, crossover, and then form the optic nerve. What you'll notice in this figure here is that information from each eye goes to the back of the brain, which is the occipital lobe. The visual cortex, where we see, is located in the occipital lobe, or the back of the brain. And what you'll also notice is that some information crosses over to the other side of the brain, while some information stays on the same side of the brain. And you'll note that the information from the right side of the visual field for each eye is going to project to the left side of the brain, and that the information from the left visual field of each eye is going to project to the right side of the brain. So the firing of the neurons from the retina to the back of the brain through the optic nerve is what's going to allow us to see those images. The brain is going to perceive those correctly, even though the image was inverted on the back of the retina. As the impulses get fired to the back of the brain, that visual cortex in the occipital lobe, the brain is going to perceive these images correctly, and we are going to be able to process that as vision and what we see. Thank you so much for watching my video. I hope that it helped you understand how we see. Obviously, sight is a little more complicated than this. I didn't go through all the biochemical pathways that occur in the rods and cones and how that signal gets sent. I will make a more in-depth video for that. This is just an overview video so that you can see how the eye works and how we see. If you have any questions or comments, please make sure to put them below. And as always, I would love your support by liking this video and subscribing my channel. Make sure that you also click on those notifications so that you never miss out on a new video. Thank you.